Today's session is the first in our research and data management series. We'll be diving into a topic that directly affects the credibility, usefulness, and impact of our research: data integrity and reproducibility. By the end of this session, I want you to walk away with a clear understanding of four things. First, what we mean by data integrity in scientific practice. Second, why reproducibility is the foundation of trustworthy research. Third, how these principles apply in your day-to-day lab work. And finally, how to recognize and avoid integrity risks that can invalidate your results or damage your reputation. Here's why this matters. A 2016 Nature survey of 1,500 scientists found that over 70% had tried and failed to reproduce another scientist's results. That's a big problem. Irreproducibility undermines public trust, slows scientific progress, and wastes funding. As John Ioannidis put it, science isn't self-correcting unless we make it so. We need to actively design our research for integrity and reproducibility. Data integrity means that your data are accurate, complete, consistent, and reliable over time. Think of it like a building. Accuracy is the foundation. Completeness is the walls. Consistency is the structure holding it all together. Timeliness and validity ensure it's fit for purpose. If any of these are compromised, your whole research structure becomes shaky. Now, in real labs, especially microbiology labs, we often encounter threats to integrity. Samples get mislabeled. Metadata like collection time or growth conditions are missing. Version control is weak, especially with code or digital files. And we've all seen image manipulations that raise ethical flags. These aren't hypothetical, they're happening in real papers. Let's clarify some terms that often get confused. Repeatability: same lab, same setup, same data, same results. Reproducibility: different lab, same data and methods, same results. Replicability: different lab, new data, same methods, similar conclusions. Example: a team published a PCR-based method to detect antibiotic resistance genes. Another lab attempted to reproduce it, but failed. Why? The annealing temperature wasn't documented, just standard conditions were written. That's a minor omission that had a major impact. Here's a real example timeline. Day 0. Sample collected from E. coli isolate. Day 1. DNA extracted, but no metadata on bacterial growth conditions. Day 2. PCR run. Annealing temp was handwritten in a lab notebook, not uploaded. Day 10. Results submitted and published. Week 3. External lab tries to replicate results and fails. Week 4. Back and forth emails reveal missing information. This is not misconduct, it's just sloppy data management. But the impact? Lost trust, wasted resources, and a stain on credibility. So what can we do? Use the right tools. Electronic lab notebooks, ELNs, to keep detailed, time-stamped records. Audit trails in LMS systems to track changes. Data versioning tools like Git or DVC for code, scripts, and datasets. And adopt SOPs and ISO standards that make your protocols bulletproof. There's also a framework around us that we need to respect. From the FELASA Animal Ethics Guidelines to ALCOA Plus Principles of Good Documentation to Publication Ethics under COPE. These aren't bureaucratic hoops, they're quality assurance. The research lifecycle, from ethical review to data archiving to publication, needs to be integrity-driven. You play a central role in building a culture of reproducibility. Log everything, even things you assume are obvious. Keep your raw and processed data safe, separate, and backed up. Share full protocols, not summaries. Encourage colleagues to verify or repeat parts of your workflow. You're not just doing this for others, you're doing it for your future self when you revisit your project 6 months later. Let's recap. Define and protect the integrity of your data from day one. Use digital tools that support reproducibility. Follow ethical and institutional guidelines. And above all, log and document everything. This isn't about perfection, it's about responsibility. If you want to go deeper, check out these. Cope for Publication Ethics. Nature's Reproducibility Series. NIH Guidelines on Rigor and Transparency. And books like The Reproducibility Crisis in Science. These resources are part of your professional toolkit. Use them. Thank you for listening. Please like, share, and subscribe for more.